All, this is Dr. Mobeen Sayed from drbeen.com. Hopefully, you are doing good. I hope that you have been enjoying the lectures or the guests that are coming on. Now we have Dr. Paul Marek, we have Dr. Pur Hassan, and yesterday Dr. Bruce Patterson. Hopefully, in the coming days, hopefully Dr. Zelenko feels well, and we'll have him. And we have a couple of more doctors who are going to join us. If you have someone that you feel should be here and have a discussion with you, please let me know in the comments, and I would reach out to them and see if they can join us. So with this, today's discussion is very important. This has been a topic for some times lately in the context of vaccines, in the context of antibodies that are fading away, in the context of T helper 1 response, in the context of the testing done. And in some people, the tests are negative and the antibodies are not produced, and they swear that we had the symptoms. And then in context of the long haulers, this discussion is the latest part of that discussion. And I wanted to go a little deeper into understanding of what determines the T helper 2 pathway or the antibody formation pathway versus T helper 1 pathway. So that is the discussion today. Now, an interesting thing is, for this specific concept, that do we go T helper 2 pathway or do we go T helper 1 pathway, we still, after decades of research, doctors have not been able to really figure out what causes one pathway to be selected versus the other one. And this tells you how interesting it is that the doctors and the scientists have been researching, and how much advancement we have done in the field of medicine, and how much still is out there that we do not know. This also tells me sometimes when people write so generously, this doctor is a quack and that doctor is a quack, and they just profusely insert them. I feel that the doctors and the researchers, we owe them a lot of respect for the work they have done, for the studies they do, for the research they do, and they have brought us this far. We will continue moving forward as well, but this work that is done is tremendous. So with this, let's start our discussion. I'm going to share my screen. So here, we have been looking at this for some time. Almost in every lecture, we talk about it. That we have, let's say over here, this is a part of the innate arm. This may be a dendritic cell, or this is a macrophage. Then it connects with the pathogen, and then a cascade of events occur which either take us to this pathway where we make antibodies, which is the path which is the pathway for the COVID-19, which is the dominant pathway that we are seeing nowadays with the COVID-19. The other possibility is that this pathway gets engaged, which is the T helper 1 pathway, or the cytotoxic pathway, which nowadays we have been getting a lots of discussions around this, that it is possible that some people who do not have antibodies have actually this pathway activated. And we talked about it before too. Yesterday, Dr. Patterson was talking about these pathways as well. So I thought we'll dig a little deeper. So the question to hold in your mind is, what is going to start one pathway or the other? So I'm going to now present various theories that are out there, which postulate that maybe one pathway is triggered by one reason or the other. None of these are actually fully finalized. None of them have become the final answer yet. So let's start from here. One of the theory is that when a innate arm cell, for example, dendritic cell or a macrophage, connects with the virus or pathogen, that may be a virus, that may be a bacteria, that may be a fungus, or that may even be a cancer cell. So anything that is harmful for us. What happens is that on a virus, let's talk about COVID-19, on the virus, there are patterns, there are structures on the virus which are unique to it, which our innate arm cells can connect with either through toll-like receptors. These are the receptors that are going to connect to one part of the virus. For example, let's say in case of COVID-19, S protein, or pattern recognition receptor. So there are many kinds of receptors on dendritic cells and the macrophages that can connect with various antigens being parts of the bad things like viruses or bacteria. So the first theory says that it depends which kind of receptor connected with the virus. So if it was a toll-like receptor that got connected with the virus or a pathogen, then the macrophage or the dendritic cell will try to trigger T helper 1 pathway, this pathway. And how would that do that? We'll look at that in a second. But the first theory that if toll-like receptors are engaged first, then the T helper 1 pathway may be selected. On the other hand, if pattern recognition receptor, which is another way to bind with the pathogen, if that gets engaged, then T helper 2 pathway may be taken. So one theory says that it really depends upon our innate arm and what kind of receptor got connected with the antigen to determine what kind of response will appear. The same theory, and I want to very quickly show you the mechanisms here as well. So if anybody wants to go in, and it's a good read, it is a little bit outdated. So if you see here, not outdated that it is incorrect, it is a little old. So this is 2008 March. Here they're talking about immunological decision making. How does the immune system decide to mount a helper T cell response? Another interesting one, if you see here, this is actually 20 June 2019. So it is more recent, but still Peter, the Dr. Brescher over here, is still discussing the same theories, and he is proposing his own theory, which is called a threshold theory, and we'll talk about it. The point I'm making is that, one, you can read this, and number two, since 2000 till now, or since 1960s till now, we are still trying to figure out what causes one pathway over the other to become active. Now, the same theory here that says that it depends upon what kind of receptor got engaged, that theory also says that it also, this decision making is also influenced by the tissue factors. So of course, when there is a pathogen somewhere in the tissue, that tissue is going to start getting damaged. So when the tissue is damaged and the local inflammation starts, there are broken down pieces of the tissue. So let's say this is a cell that got broken down. So the necrotic cell or broken down cells lipids are influencers as well. The local cytokines released by the inflammatory process that just started, these are influencers as well. For example, heat shock proteins or tumor necrosis factor or other interleukins that are either released from the damaged tissue or released from the local immune cell. All of those together, so let's say these are the tissue factors that are connected on the dendritic cell or macrophage. So now what kind of receptor got engaged with the pathogen, and then what kind of tissue factors were available when the decision was to be made that we're going to go T helper 1 pathway or T helper 2 pathway, that determines what happens in this area. And now we have talked about this area a lot as well. This is a naive T cell. A naive T cell does not know that is it going to become a T helper 1 cell or T helper 2 cell. It can become either of them. So when it connects with the macrophage or dendritic cell, as I have been saying before, there are going to be co-stimulations, right? So there are going to be hugs and kisses. And a couple of lectures ago, we actually went into a detail of all of those co-stimulatory models, including molecules and cytokines. Over here, I would just say this much, that depending upon the type of T cell receptor, depending upon the cytokines available, we know that the pathway is going to be T helper 2 or T helper 1. Now, the cytokines that are going to be available here are dependent upon these macrophages or dendritic cells, which connected with the pathogen in the first place. So it is their duty, it is the first responder's duty to provide a correct type of cytokine, which will then allow T helper 2 or T helper 1 pathway to be taken. There is a problem in this theory, and the problem is what tells the macrophage or a dendritic cell to decide that a humoral response means the response with the antibodies, B cell response, T helper 2 pathway response. A humoral response will be better over a cytotoxic response. And just to give you a quick idea, we know that humoral response, it is called humoral because the antibodies that are produced are released into the tissue and in blood. And so blood use is called humor. So when the blood receives the antibodies, then the antibodies travel in the blood, they go everywhere around, and then they try to take care of the pathogen wherever it is located. This is why we call it humoral response. On the other hand, this response where the cytotoxic T cell or CTL becomes active. This is a cell that Dr. Patterson yesterday was calling CD8, which is correct. It is a CD8 type cell. It is also, when he was talking about it, he was also calling it an effector cell. Both of these cells, the plasma cells that are making antibodies or the T cells that are going to kill the other cells, are called effector cells. They are the final cells that would make the effect. So the question is, what tells the macrophage or dendritic cell to decide to release what kind of cytokines to then influence the pathway? That is not known. We do not know it. And I'm going to share one more theory. So the first theory now we are, I hope, good with, and that theory is it is the macrophage. It is a type of receptor that is engaged on the macrophage. It is the local tissue factors that are available from the damage that is occurring around them. That in turn allows the macrophages to release specific kinds of cytokines which would take the pathway one way or the other. So one theory done. Any issue with this theory? Of course, it has its own weaknesses and strengths, but this is a theory. Second theory, which is also very, very important, and this is going to be important in case of when we are wondering that what would happen with vaccine, for example, what should be the right vaccine dose. And similarly, when we wonder that why did one person's immune system responded with the humoral response versus the other immune system responded with the cytotoxic response. So there is one more theory here, and that is called the threshold theory. And this is this one, 5 June 2019. This has been given before as well. It's not a new theory, but over here, the Dr. Brescher was saying that we should look more into this theory and use this theory as the possible science instead of the macrophages. So what is this theory? This theory says that the amount of antigen present in this interaction of a macrophage with a naive T cell, that determines what will be the pathway. So he said that if the viral load, let's say, or the antigen quantity is less, then usually there is no response. Fine, makes sense. If the antigen's amount, the viral load is intermediate, then the T helper 2 pathway is taken, which is humoral pathway. So that means in those folks who have antibody production, probably their immune system had a lesser amount of viral load or intermediate amount of viral load, and that presence of that intermediate quantity of the antigen caused the T helper 2 pathway to be selected. The third part is if the viral load is too high, then the T helper 1, or I shouldn't say viral load, if the antigen quantity is too high, may that be from a virus or a bacteria or a fungus or a cancer cell or a broken cell, whatever that is. If the amount of antigen is too high, then T helper pathway 1 is selected. What does this mean? This means, and so let me, before I talk about what does this mean. So what they're saying is in this theory that this naive T cell, it would go T helper 2 or T helper 1 cell route based on what kind of, how much of the antigen was presented to it. Now, what does this mean? What this means is that, let's say we are talking about vaccines. It would really depend upon the dose of the vaccine to decide what kind of immune system arm will become active. That means it is not necessary that we only look at the vaccine dose to say we want antibodies. Maybe we want cytotoxic T cell response and we don't want antibodies. Maybe that is the right path. And because people have been responding with the antibody response more often, maybe that is not the right response. We don't know yet. Just for a reminder, we had done this discussion a few days ago that what we saw was in communities, in various people in the same community, we saw that there were children who have a totally different kind of a response. Then there are folks in the community that have T helper 2 response or antibody response that we talk most often. Then there are folks who develop T cell response or the cytotoxic T cell response. And then there are folks who do not produce any response at all. So what is the right response in here? Which response should a vaccine trigger? Or which response, when the virus has triggered, is the correct response? What will prevent the cytokine storm versus not is not yet known. But this theory says it depends upon the amount of antigen. So this is the threshold theory of the antigen. Then moving on to the next part of the discussion, same theories are we working with. That is, this is also known. It is known. So the observation is there, the evidence is there. Why does it happen is not known. It is known that when a naive T cell is connected with a macrophage and has an antigen presented, then it depends what kind of cytokine is released in the environment at that time. If interleukin 12 is released, then the naive T cell preferably goes T helper 1 pathway. If interleukin 4 is released, then the naive T cell would go T helper 2 pathway. What does that mean for us? That means that, for example, if we want someone to develop immunity and we found out that T helper 1 pathway, the cytotoxic pathway is better, let's say for COVID, then not only we should give the vaccine, we should also make sure that interleukin 12 is in increased amount so that the pathways can move towards T helper 1. On the other hand, let's say if we determined that the antibody response, the humoral response is the better response, then as we give the vaccine or as the person encounters the virus, we have to make sure that they have interleukin 4 in their environment as well. And the cells that generally produce these interleukins, in case of interleukin 12, we know that macrophages, they produce this. So this macrophage that was connected, or dendritic cell that was connected with the pathogen, will produce interleukin 12. We know the source here. For the interleukin 4, we do not know the source. When I was in medical college, we used to talk about it that we don't know where this interleukin 4 comes from. Even today, we only have speculations. We do not know where it comes from and drives the immune pathway in T helper 2 side. The cells that are known to produce interleukin 4 are natural killer cells, eosinophils, and mast cells. So maybe one of them or all of them or some of them are producing interleukin 4. And maybe that is by intent, or maybe that is accidental. Maybe this was just that interleukin 4 was present at that time and the pathway went to T helper 2. So this is the threshold theory. Then if we continue on to the pathway once more, next step here. Once a specific type of cell becomes active, that is, for example, T helper 2 pathway becomes active, or T helper 1 pathway becomes active, then these cells release interleukins that would suppress the other side. So that means they would continue to amplify their own wing. They would continue to strengthen this pathway then by releasing interleukins or cytokines that would amplify the cells in this pathway. On the other hand, let's say in somebody, T helper 1 pathway became activated. How did it become activated? If you look at the theories, maybe the antigen quantity was more, maybe the interleukin 12 was present at that time, maybe the TLR got connected with the pathogen instead of PRR. Whatever was the reason, eventually we are in this pathway, which is T helper 1 pathway. And if that is the case, now the interleukins that are going to be produced from here, for example, interferon gamma, for example, tumor necrosis factor, interleukin 2, and so on, they are going to try to keep this pathway working and suppress the other side. So this arm will become stronger. So once we are in this stage, then the cells try to keep themselves strong and suppress the other cells. Continuing on with the last theory for what causes the selection, this is called the eradication theory. Eradication theory says that when a pathogen is present, let's say COVID-19, and the cell's humoral side or the cytotoxic side, these cells get connected with the pathogen. For example, over here, you are seeing this is a cytotoxic T cell. This is a cytotoxic T cell. It is connected with a tissue cell. Let's say in this case, this is a lung epithelial cell. And the epithelial cell is presenting the antigen to it. And they're connected. Maybe one cell type gets to connect more than the other cell type. These cells also have receptors to bind with the antigen. So let me just quickly make that. So let's say this is a receptor here that also allows the B cell to bind with the antigen as well. Let's say in this case, COVID-19. So let's say the virus comes into our body and the cells are now connecting with it. It may be that the T helper 1 pathway, the cytotoxic pathway, the cells there are more connected with higher concentrations. And as soon as the signal comes that, all right, start working, they become more active and they start functioning and the other side gets eradicated. Or maybe that the B cells are connected more, they are exposed more to the antigen, the virus. The T cytotoxic cells are exposed less to the virus. And as soon as a signal comes to say, all right, start functioning, B cell get up and start running faster than the T cell, cytotoxic T cell, and the result is cytotoxic T cell wing becomes eradicated. We don't know really what happens. There is another theory, and that theory actually starts from here, where what they say is that this macrophage here, which is influencing this pathway one way or the other, this macrophage itself is influenced by these cells. The short of this all is we have no idea what starts the T helper 1 cell pathway versus the T helper 2 pathway. We know some things. The known things are interleukin 4 presence makes the pathway goes toward T helper 2 pathway. Interleukin 12 presence make the pathways go toward T helper 1 pathway. Presence of tissue factors and engagement of various receptors can alter the pathways as well. Most strong theory is it is the dose of the pathogen or the antigen that determines which pathway is taken. Now, the final question I'm going to ask or answer, that question is that fine, this is what happened when the pathogen got exposed to us for the first time, when it arrived in our body for the first time. What happens when there is a re-exposure? What happens when our immune system sees that pathogen again? Will now this immune system behave differently? Maybe the first time when the pathogen arrived, it connected with the TLR, and that caused it T helper 1. Maybe the next time when it arrives in our body, it connects with PRR, and now it causes the T helper 2 pathway. What happens is, interestingly, our immune system keeps copies of those cells that had become active the first time. So these are the memory cells. So because the memory cells are already present, as soon as the pathogen arrives in our body once again, these memory cells very quickly connect with the pathogen and start responding and start producing interleukins that would keep their pathway intact. So the whole process of starting and determining the pathway does not occur the next time, or it is weaker because the previous memory cells have already started functioning and already started to strengthening their own site. This is why when the reinfection occurs, the pathogen is very quickly taken care of by the immune cell with the similar response as there was the response the first time around. So this is the discussion. It has implications that do we go T helper 1 pathway or T helper 2 pathway? Do we prefer one over the other? And what causes the preference to be there? And what would happen with the vaccines? Will the doses be enough to cause, let's say, T helper 2 pathway or T helper 1? Should we have T helper 2 pathway or should we have T helper 1? Then similarly, the tests, they are looking at antibodies. Should we start looking at T cell receptors as well? And similarly, the herd immunity calculations, they are also looking at antibodies. Should we start looking at T helper T cytotoxic T response as well? Then one bigger question that still needs to be answered is the T helper 2 pathway, humoral pathway, a better pathway, or T helper 1 pathway is better pathway in terms of preventing the cytokine storm? Is it possible that T helper 1 pathway, the cytotoxic T cell pathway, does not end up in the cytokine storm and T helper 2 pathway ends up in that storm? We don't know yet. But this needs to be looked at as well. So this is a discussion. One last part I wanted to share with you, and I think that is very exciting as well. This is a new study or new discovery that just came out, I think a day ago or so. This is a discovery which said, how does a killer T cell become active? What does that mean? This T cell, cytotoxic T cell, how does it actually become cytotoxic? How does it become active and start functioning? And what they said was, the researcher, they said that in the cytotoxic T cell, when it connects with the antigen presenting cell, so we're talking about CD8 cell or the cytotoxic T cell, when it connects there with the antigen presenting cell, the cytotoxic T cell expresses a molecule called RK motif, which is usually not expressed. RK motif is usually hidden, but as soon as the antigen connection occurs, then this motif becomes expressed, which then causes co-stimulation, which then causes the cytotoxic T cell to become active. This is a new discovery. This discovery had never happened before. And so we did not know that how does it just start killing other cells? What makes it active? And that is the stimulation of the RK motif. What does it mean generally from clinical point of view? That means tomorrow there can be drugs that can selectively either activate the RK motif, or they can block the RK motif and then cause either the suppression of the cytotoxic response in certain autoimmune diseases or improvement of the cytotoxic response in certain other infective diseases. So this is what we have for today. Thank you very much for joining. I hope that you are enjoying these talks. Please do me a favor. Please like, subscribe, and share. Please stay safe and healthy, and we'll continue tomorrow. Thank you.